In this video, we will discuss about how this STUB1 along with this IFN gamma in CD8 plus T cells can delay tumor growth in the tumor microenvironment. So, how they come up to this hypothesis? So, in the previous video, we have understood that STUB1 plays a role towards this anti-tumor response in the CD8 plus T cells, and once this STUB1 has been knocked down, then they have seen the increased CD8 plus T response against the tumor growth rate that can be seen in this panel J clearly. And now they wanted to check whether STUB1 plays the same role in the tumor microenvironment or not. So in order to check that, what they did is they checked the tumors or cells from the TDLN and tumor sites, and they check it for the SLAM F6 positives and TM3 positive cells. So in the case of control, they can see that there is SLAM positive cells, uh, and this TIM1 negative cells has gone up, or we can this SLAM positive cells and TIM1 negative cells, that is, it has gone down in the case of STUB1 knockout. So this differentiation towards this TIM1 positive seems to increase as this TIM1 positive cells has gone up in the STUB1 knockout compared to the control. However, when they check it for the tumors, here they have find out that the amount of cells in the case of this SLAM F6 negative are increased compared to the control case. So this indicates that the cells behave slightly differently in the TDLL and tumor site. However, their per cell killing capacities does not get affected. So and they have also shown that the total number of CD8 plus T cells in different populations remains same. For example, in the panel B, we can see that SLAM6 positive and TIM3 negative, and SLAM6 positive and first SLAM6 positive and TIM3 negative remains same in the control and STUB1 knockout, and for the case of a SLAM6 positive and TIM3 positive, they also remain same. And however, for the SLAM6 negative and TIM3 positive, they can see that SLAM6 negative and TIM3 positive, so that is these populations. This is non-significant in TDLN and tumor cases, and all other cases it's significant because the Q value is 0.002 and 0.002 in both the cases, indicates that these cells behave differently in the TDLN and tumor side when they are STUB1 knockout. And this is in terms of the percentage, and in terms of the total number of populations, it has also shown that in case of total number of CD8 plus T cells populations, it is not statistically significant. As for the case of SLAM6 positive and TIM3 negative, the p values is 0.6, and but for the SLAM6 positive and TIM3 positive, both are positive, that is these populations, it is statistically different, that is in the STUB1 knockout these populations has rises that can be seen here, and the SLAM6 negative and TIM3 positive at these populations is also statistically significant, that is p value is less than 0.05. And for the case of this tumor, these are the panel B is for this TDLN part, and panel C is for the tumor part. So in the tumor part, we can say that the populations are high in the case of this SLAM6 negative and TIM3 positive, that is these populations, and here the Q value is also statistically significant for all the three cases, that is the as a STUB1 knockout affect the cell morphology or differentiation significantly. And in terms of the total number of cells also, we have seen that in all the three cases Q value is less than 0.05. So indicates that there is there is a total number of cells, or indicates that the differentiation occurs differently in the case of STUB1 knockout compared to the control case. Now they wanted to, since they have now find out that differentiations varies in the STUB1 knockout, and or in the case of the STUB1 is present in the CD8 plus cells, they wanted to check some other factors, such as so they check it for the perforin enzyme, which is the cytokines for triggering apoptosis in the tumor cells, and which are usually released by the immune cells. So they check it. In the case of control, they have seen the granzyme positive and perforin positives around 77%, but when in the case of STUB1 knockout, this has gone up to 95.6%. So this indicates that the cytokines that are released by the STUB1 knockout cells is further accelerated. And one more thing that they have not presented in the main figure is about they did the RNA-seq analysis of this STUB1 data and control data from TDLN and the tumors, and they are able to find out that most of the genes that belongs to the T cells activations are upregulated in the case of STUB1 knockout. So that's how they are checking these genes, which are also upregulated in the case of STUB1 knockout, that is granzyme and perforin. So they are high in the case of STUB1 knockout, and we can see that p-value is around 0.0058. And they also check it for IFN gamma, and they can see that IFN gamma also increases in the STUB1 knockout. And these quantifications are shown in the panel E, where we can see that in the case of STUB1 knockout, the IFN gamma is high, indicating that the cells are differentiating towards cells in which STUB1 knockout is there are more prone towards these activations compared to the case where STUB1 is present. Next, they did the in vivo studies where they transduce the STUB1 knockout cells in the wild type and or in the IFN gamma or in the perforin. And for the control case, they transduce these wild type cells with control guide RNAs, and they transduce them in these B16 OVA tumors and monitor the growth rate to check whether it will this IFN gamma or perforin also has an effect on the tumor growth rate or not. So in the wild type case, we can see that the tumor growth rate increases has a significant pace, and it is comparable to the case where IFN gamma knockout is there and STUB1 knockout is also there. But for the case where wild type, for the case where only STUB1 knockout is there, they see a delayed tumor growth, indicating that this IFN gamma also plays a role towards suppression of the tumor growth. But for the perforin, the results are not shown here, but it is in the supplemental figures. They see that, or they have observed that there is no tumor delayed occurs, or it is not working towards delaying the tumor growth rate. And in terms of the survival rate also, we can see that when only the STUB1 knockout is there, they are able to see higher survival rate compared to the case where IFN gamma and STUB1 both are knockout, or the wild type case where both are present. So this comes to a point that STUB1 has a negative role that they have find out in the initial studies, and STUB1 knockout will promote CD8 plus cell efficiencies, whereas IFN gamma has a positive role. Thereby, IFN gamma knockout will suppress the tumor or CD8 plus T cells activity. So when the STUB1 knockout is there and IFN gamma is present, at that time they are able to see high survival rate and delayed tumor growth. So this is how they validate their hypothesis that STUB1 along with IFN gamma in the CD8 plus T cells can delay the tumor growth in the tumor microenvironment conditions. I hope you understood some of the results that are presented in this figure. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.